connaissance,
recherche,
sciences,
et sciences mathématiques,
théorie,
physique,
philosophie,
conférence,
archéologie,
environnement,
nature.
I'm going to talk about factors explaining bone histological variation with a special emphasis on resting metabolic rates. I arrived to the laboratory of Armand de Ricqlès 12 years ago as a postdoctoral student with the aim of quantifying these factors explaining bone histological variation. By this time, papers dealing with this issue used a qualitative approach, which has produced valuable interpretations. However, it involves a risk of lack of repeatability. Thus, we developed quantitative statistical methods to avoid this problem. One of these methods was variation partitioning. Is a method which considered that, for instance, this rectangle encompasses all variation of a given bone histological character. We focused our attention in a first time on the historical component of variation, which corresponds to the fraction of variation explained by phylogeny. We used regressions on distance matrices, and our results show that the phylogenetic signal depends, the phylogenetic signal for a given character depends on the taxonomic level. For instance, for vascular density, you can see here that the phylogenetic signal is different for the different phylogenetic levels. I would like to note that these values, we know now that these values are underestimations because we know that the regression on distance matrices underestimate the percentage of variation explained by the independent variable. Thus, we should see this graph as conservative values. The phylogenetic signal is greater than what we can see. Afterwards, we were interested on functional component, and in a first time, we focused our attention on the biomechanical aspects. More precisely, the effect of the frequency of cyclical deformation on bone structure. It has been shown that flight involves cyclical deformations in the furcula of birds. We can see here that in every flight cycle, a big deformation in the furculae occurs. Our hypothesis is that these cyclical deformations produce microcracks that we can see here that are repaired by secondary osteons or Haversian bone. Well, to test this hypothesis, we made a comparative study in a phylogenetic context, and in fact, we analyzed the structure of the furculae of a sample of birds, which included 4 clades showing a high-frequency flapping flight. Anatidae here, Rallidae here, Alcidae, and Podicipedidae. our results show that the density of secondary osteons was significantly higher in clades showing high-frequency flapping flight than in their respective sister groups. A second functional factor explaining bone histological variation is bone growth rate. Leticia Montes has shown this morning that bone histological diversity, the rectangle here, is significantly explained by bone growth rate. Well, I will pass quickly. Lepidosaurs and chelonians show parallel fibered bone formed at low growth rates. In crocodiles, accretional growth is achieved not only at the periphery, but also inside large cavities, here included in bone cortex, called primary osteons. Finally, in mammals and birds, an initial scaffolding is laid down at high to very high growth rates, and later on it is filed later on. Using phylogenetically independent contrast, we have shown that a significant portion of bone vascular density is explained by bone growth rate. But we can wonder about the determinism of these bone growth rates. In fact, building new tissues at high growth rates involves high rates of protein synthesis and degradation, a high protein turnover, which is very energy demanding. Thus, resting metabolic rate may be a proximal or mechanistic factor explaining limiting the maximum growth rate. Thus, we performed a quantitative study to test this hypothesis. Resting metabolic rates were quantified using respirometry, and we were able to show that there is a direct linear relationship between the resting metabolic rate and the bone growth rate. Well, until recently, the analysis of variation of morphological characters, not only bone histological ones, but in general bone morphological characters, and the interpretation of their significance was remained within either the functional or the historical approaches, taken separately depending on each worker intellectual tradition. For systematics, for instance, the functional adaptation to current conditions, the autapomorphism, may mask a pure phylogenetic signal. In contrast, for functional morphologists, phylogenetic patterns are constraints that may prevent organisms from reaching optimal adaptation to current conditions. From our point of view, the dichotomy between functionalism and historicism is misleading, because any biological feature has at the same time a functional significance and it contains a phylogenetic signal. Our variation partitioning approach allows us to quantify this overlapping, which is known as a phylogenetically structured functional variation. Well, remember that this rectangle encompasses the variation of a given histological character. After explaining the functional and the historical components, there is a big quantity of variation which is unexplained by these factors. We can wonder about other factors able to explain this unexplained variation. Architectural or structural factors evoked by cell care, like the properties inherent to the materials with which organs are constructed, could explain a part of this character variation. We published 2 years ago a paper allowing to take into account this third group of explanatory factors. Well, I will show one of these structural or architectural factors linked to the effect, the size effect on bone histological characters. Osteocytes here communicate among them and with the endosteum here and the periosteum there through canaliculi. You can see here canaliculi between the periosteum here and the osteocyte, and here canaliculi between the endosteum here and the osteocyte. When bones are small enough, osteocytes get oxygen and nutrients directly from the endosteum and the periosteum, because the ratio between the surface of endosteum and periosteum and the volume of bone tissue is big enough. For instance, the femur of Sturnus here has a surface to volume ratio of almost 9 and lacks vascularization. And you can see here that all osteocytes in this bone obtain the oxygen and nutrients directly from endosteum and periosteum. As bone size increases, as we can see here in buteo, the ratio surface to volume decreases, and vascularization becomes a structural constraint. And we can see here that with a ratio of almost 4, well-developed vascularization appears. And this fact is interpreted here as a structural constraint that can be put here. Well, a quantitative analysis in a sample of sauropsids shows that using phylogenetically independent contrast shows that there is a significant relationship between bone size and bone vascular density. Well, until now, we have used regression analysis to explain the variation of dependent variables, here bone tissue, using 3 kinds of explanatory factors: historical, functional, and structural. Now, we will use this regression analysis to estimate the functional features of extinct organisms using predictive models, which are constructed with extant organisms. Well, we have shown that there is a relationship between resting metabolic rate, which limits bone growth rate, and between bone growth rate and bone microstructure. Then we can use bone microstructure to estimate both bone growth rate and resting metabolic rate. We will focus here on the estimation of resting metabolic rate, but to make reliable predictions, we should control for at least size, skeletal element, ontogenetic stage, developmental mode within the altricial precocial spectrum, and phylogeny, what we did in our analysis. I don't have time to explain all the steps. I will show you the results. For extant species, we quantitatively measured the resting metabolic rate. For extinct ones, we used the predictive model, and I would like to thank here Michel Laurent, who developed a method to estimate confidence intervals for internal nodes. He published this method in his paper on Cope's law. And with this method, we were able to estimate not only the value estimated for the last common ancestor of different nodes, but also confidence intervals. You can see that the condition estimated for the last common ancestor of diapsids involves a low resting metabolic rate, that this primitive condition decreased in Pseudosuchian, taxa more closely related to crocodiles than to birds, although this reduction wasn't statistically significant. In contrast, in dinosaurs acquired higher resting metabolic rates than the last common ancestor of diapsids. Note that the difference between the value estimated for the last common ancestor of Allosaurus and extant birds is significantly higher than that estimated for the last common ancestor of diapsids. Well, in conclusion, bone histology contains information useful to estimate resting metabolic rates, and new statistical methods in a phylogenetic framework allows us to make reliable estimations. Thank you for your attention.